Transport of chemicals across the plasma membrane is required for normal cell functions. Some chemicals, like oxygen, are imported to maintain metabolic processes in the cell. Others, like carbon dioxide, are exported as the cell produces them. Communication occurs between cells when substances produced by one cell, such as neurotransmitters, induce the transport of chemicals across the plasma membrane of another cell, making the generation of action potentials possible. And that is something we will be studying in chapter 12. Some chemicals pass through the membrane boundary directly through the phospholipid bilayer. Others can only pass through the membrane with the help of a transport protein. Let's explore that. There are two major types of transport: passive and active. Chemicals that are transported by passive processes travel down their concentration gradient. In other words, the chemicals flow from high levels of concentration to low levels of concentration. Active transport moves chemicals against or up their concentration gradient and requires energy in the form of ATP to do so. And here we have a demonstration of the passage of ions or molecules either against the concentration gradient, which is up in this case of sodium now, or down the concentration gradient when the ion channel simply open and then diffuse through. Passive transport processes are driven by concentration gradients. They include diffusion through the bilayer, diffusion through membrane pores or gated channels, facilitated diffusion using transport membranes or proteins, and finally osmosis. Active transport processes use energy from ATP to move molecules. They include transport of chemicals across the membrane using protein pumps and transport a large volumes of fluid, large particles, or cells via transport vesicles. With passive processes, gases and nonpolar molecules pass through the bilayer along their concentration gradient. In the process of diffusion, ions and glucose cannot pass through the phospholipid bilayer. Charged molecules, like ions, diffuse to the plasma membrane via membrane proteins. leakage pores are always open. In other words, these are sodium and potassium leakage channels. The number of pores or channels determines the amount of ions that diffuse across the membrane. Gated channels are opened when they bond to chemicals or experience changes in voltage. And again, this will be explored later on in great detail in another chapter. Molecules like glucose bind with the transport proteins that change shape to facilitate the transport of molecule. One of the roles of the hormone insulin is to promote production of transport proteins. Water passes in and out of the cell primarily through pores called aquaporins. During osmosis, water, like ions, will move down its own concentration gradient. The concentration gradient of water is to a large degree determined by the fluid's solute concentration. The direction of osmosis depends on relative concentration of solutes in the cytosol and extracellular fluid. If the ion concentration is the same on both sides, then the water moves in equal amounts in both directions. A hypertonic solution has a low concentration of water, so the direction of osmosis will be toward the solution. A hypotonic solution has a high concentration of water, so the direction of osmosis will be always from this solution. Primary active transport or of charged or polar solutes requires membrane proteins or protein pumps that use energy supplied by ATP in order to work. This causes a conformational change, moving the molecule up its concentration gradient. The sodium-potassium pump is an example of a pump used to maintain electrochemical gradient in neurons. Actively transporting ions is the major way the cell has of maintaining water balance. Secondary active transport occurs when the concentration gradient of sodium or hydrogen ions produced by primary active transport drives the transport of another chemical. Low cytosolic calcium levels are maintained by secondary active transport. Another Example of secondary transport is the absorption of nutrients from the digestive tract into intestinal cell. Endocytosis is the movement of large particles of macromolecules into the cell. During endocytosis, a small round sac called a vesicle is formed from an existing membrane and used to transport or for transport. Three types of endocytosis are phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis is a common form of endocytosis and part of a vital defense system protecting the body from disease. Large particles like bacteria bind to a receptor, causing the membrane to extend to form pseudopods, which then surround the bacteria, forming a vesicle called a phagosome. The phagosome will fuse with the lysosome, which contains digestive enzymes, and enzymes in the lysosome will digest the bacteria. Pinocytosis is the process in which cell drinks a tiny droplet of extracellular fluid, including solutes. The plasma membrane folds inward to form the vesicle, in which then pinches off and moves into the cytosol. Like phagocytosis, vesicles fuse with lysosomes and the solutes are digested. Finally, in receptor-mediated endocytosis, material is imported like certain hormones, antibodies, vitamins, and low-density lipoproteins into the cell. Specific receptor proteins bind with these substances and then interact with the peripheral proteins called clathrins found on the cytoplasmic side of the membrane. And so that area of the membrane is called clathrin-coated pit. When the coated pit and receptor interact, the membrane folds inward to form a vesicle surrounding the substance and receptor. Once inside the cytosol, the vesicle loses its clathrin, which is recycled back to the membrane. The uncoated vesicle now fuses with the vesicle called endocell, where the substance is going to and the receptor are separated and sorted out. Many of the receptors leave the endosome via a carrier vesicle and return to a plasma membrane. The ingested substance and some receptors are transported in carrier vesicles to a large or to a late endosome, which binds with lysosome, where the substance is digested into usable products by cells. Transcytosis is a process common in endothelial cells of blood vessels. In this process, the carrier vesicle transport substances from the blood across the cell where it releases its content into extracellular fluid of the interstitial fluid. Exocytosis is another method of vesicular transport that moves large volumes of fluid or chemicals out of a cell. Vesicles enclosing substances for release seal to plasma membranes, and the content are exported out of the cell. Exocytosis is the way cell ejects waste products resulting from endocytosis. Nervous cells release neurotransmitters via exocytosis, and secretory cells secrete imported substances like digestive enzymes and hormones.